Good morning to everyone. Myself, Dr. Vijayakumar, Professor of Physiology. Today, I am going to handle the regulation of respiration. The regulation of respiration in human is a very interesting one. When the respiration shows any changes, automatically from our brain, there are several centers are there to control the respiration. Okay, there are several types of regulation: nervous regulation, chemical regulation, and others. Here, the main regulations by the brain are pons and medulla. So, the nervous regulations will be controlled by pons and medullary centers in the midbrain. This picture shows the respiratory center, parts of the respiratory system, and related to the respiratory sensor in the heart, and where the changes takes place while the need of the changes in the respiration. Okay, these are chemical regulation pictures shown in this, and when there is a changes in the oxygen, partial pressure, or carbon dioxide, or changes in the hydrogen concentration, there will be a reaching of impulses to the higher centers, which can control voluntarily to the respiratory system. Okay, under the nervous regulation, the first centers are known as higher centers, which is nothing but cerebral cortex. The cerebral cortex, which will control the respiration whenever it goes high or low. It will retain the respiration back to the normal within the brief period of time rapidly, and then it will escape from the effect quickly. What are the respiratory centers? One is the higher centers, I already told, cerebral cortex and hypothalamus and limbic system. These three higher centers will control or alter the respiration rapidly, but only a brief period of time, and it will escape from the effect. Next to that, medullary center and Fontaine center. There are four: inspiratory center and expiratory center. Inspiratory center is located in the medulla, known as ventral respiratory group and dorsal respiratory group. And Fontaine centers are pneumotaxic center and epimistic center, which is located just above the medulla. And main control points are, as usual, I told, brainstem controlling center, central chemoreceptor, and peripheral chemoreceptor, lung J receptor. All this come under the nervous regulation. And another point of controlling the respirations are rhythmic muscular activity and regulated by the partial pressure of carbon dioxide, hydrogen ion, and partial pressure of oxygen. The goals of the regulation of respiration is optimize muscular work done and maintain the blood gases and maintenance of the pH. Now, coming to the brain control centers, that is a ablation or a transection and electrical study, as well as the motor nerve to the respiratory muscles. In the previous picture, you can see the respiratory muscles, which is shown in the right bottom, the ribs, and in between the ribs, there are the respiratory muscles, and nothing but intercostal muscle, external intercostal muscle, and the internal intercostal muscles, as in the major or principal muscles that called diaphragm. Okay, that is the right bottom picture, shows directly innervated by the nerves and directly controlled by the brain. If any changes in the or alteration in the respiration, brain immediately will take responsibility to control the respiration back to normal. And the main controls. Now coming to the medulla and pons, which will use the efferents through the anterior white column tract and anterior cells of the pre-neck now at the level of cervical third and the cervical fifth one. And second intercostals T1 to T12. That is the thoracic segment of the spinal cord level T1 to T12. And this both will shows the changes in the inspiration as well as the expiration, nothing but the simultaneous. And this is the long inspiration due to obviously hearing river reflexes. Okay, what is hearing river reflexes? I will explain in detail after completing some explanation or some regulations. So the left side picture shows again the centers of the respiration are known as BRG, ventral respiratory group, DRG, dorsal respiratory group. The VRG located ventrally to the medulla and dorsal group located dorsally to the medulla. And this is the clear pictures of the respiratory center. There are 4 centers. The top one is called the pneumotoxic center. And the signals releasing from the pneumotoxic center always will be inhibitory. So that the mark is given as a negative, minus or negative symbol. And that the signals, inhibitory signal always goes to the agnostic center, which is a positive center. That means agnostic center alone can switch on the respiration, but a pneumotoxic center will always use the inhibitory action on the agnostic center, so that the agnostic center may switch off the respiration. That is decreasing the or controlling the respiration. And the functions of each center mentioned in the picture itself. The ventral respiratory group, which is located ventrally to the medulla, the bigger one, right side below one, so ventral respiratory group, believed to causes the process of inspiration as well as the expiration, both. And dorsal respiratory group, which is to the left side of the slides or picture, dorsal respiratory group, which will believe to cause the inspiration. So all this controlled and influenced with the nerves, vagus as well as the glossopharyngeal nerve. Okay, now centers in the medulla oblongata. Number 1, pre-Purkinje complex, that is in the nucleus of tractus solitarius, and respiratory control pattern RCPG, respiratory control pattern as present in the medulla oblongata, and parts of the DRG, I already told DRG located dorsally to the medulla oblongata, and as well as the pacemaker of the inspiratory activity and irregular gasping breathing effort. Finally, signals that project to the dorsal respiratory group. Okay, dorsal respiratory group located in the nucleus of tractus solitarius near the nucleus ambiguous. The nucleus tractus solitarius gives the sensory input from the cranial nerves 9 and 10, which leads to the cardio-inhibitory reflexes. RCPG modified to the ramp signal. That is a pattern of normal respiratory movement is called ramp signal, also called the normal respiratory pattern is called ramp manner. The frequency increased to the 1 to 5 to 2.5 seconds, and it will stop for the 2.5 to 3 seconds. So this is the normal respiratory ramp. Also it is called ramp manner. Motor output, opposite to the pre-neck nerve, inspiratory nerves of the VRG and pneumotoxic centers. Okay, ventral respiratory group, which is located lateral to the DRG, and 2 co-joined or conjoined portions, and inspiratory as well as the expiratory nerves are present, and it is located in the nucleus ambiguous. Ipsilateral laryngeal muscles via vagus, mainly it causes the inspiration. So ventral respiratory muscle believed to causes the inspiration mainly, as well as the expiration previously told. So some extent it will give the expiration also, and nucleus of retroambiguous, intercostal muscles and abdominal muscle diaphragm, mainly expiration. See, ventral respiratory group believed to causes both inspiration as well as expiration. And it will be inactive at the time of rest and active only forced ventilation. So when the hyperventilation happens, this center become activates and acts accordingly. Primary influences on the expiratory muscles, that is a forceful expiration, abdominal muscles. So these are the roles of the ventral respiratory group, which is located ventrally to medulla and lateral to the dorsal respiratory group, two conjoined portions. Okay, centers in the pons. I already told that two centers are there in the pons, pneumotaxic center and apneustic center. So the pneumotaxic center is located in the upper part of the pons, that is in the nucleus of parabrachialis, and it projects to the dorsal respiratory group, which will causes the inspiration plus expiration. Stimulation of these centers switches the phases of the breathing. That means it initiates the inspiration and expiration, increasing the discharge during the inspiration and shut off the signals for inspiration. That is the major functions of the pneumotaxic center. Normally, it will be inhibitory in nature. Therefore, when it will switch on when the breathing is behind the limit and increasing the charge during the inspiration. Finally, it will switch off the signal for inspiration. So for this, when it gets switched off, when the increased signal strength that causes the decreased time for inspiration and expiration, and increase the rate of the respiration and decrease depth of the breathing. So without depth, without deep breathing, and there is no gap between the inspiration and expiration. Therefore, these centers will take charge and release the inhibitory impulses to the apneustic center. Finally, the inspiration will be switched off or shut off. Okay, apneustic center, lower pons. Still, the functions of the apneustic center clearly not known, but believed to cause switch on and off system on the respiration, stimulation of the apneus, long deep breath, and inhibits the pneumotaxic center, stimulate the dorsal respiratory group, and it promotes the inflation of the lungs, leads to the entry of air inside the lung, the process called inspiration, inhaling of the air, and which will enters the lung will inflate, air enters in the lung, leads to the inspiration will be succeeded. Shut off for the expiration. So apneustic center normally will switch off for the expiration, and paradoxical inspiration signals of first breath. These are the major roles of the apneustic center, where it's located in the pons and perform its functions based on the changes that occurs in the lower respiratory respiratory alterations happen in the lungs. Next, pneumotaxic center, apneustic center, ventral respiratory group, which causes inspiration and expirations, and pre-Pursinger syndrome, that is a RCPG, DRG, which is responsible for the ramp signal. I already told, regular pattern of the respiration called as ramp signal. All this controlled by through the phrenic nerve, then to the inspiratory muscles, and from the vagal afferents. When the respiratory muscle gets stretched, impulses arises from the vagus and reaches the pneumotaxic center, as well as actually excitatory impulse reaches the pneumotaxic center, inhibitory impulse goes to the apneustic center. Further, apneustic center will inhibit the pneumotaxic center. Therefore, when the pneumatic center is free from the stimulations from MNC center, will exhibit its inhibitory action. Okay. Now going to the next chemoreceptors, which is the nothing but chemical receptors like increase hydrogen ion, increase carbon dioxide, or decrease carbon dioxide, or decrease oxygen, may sensitize the medulla oblongata. Accordingly, the medulla oblongata, there is the centers are present in the medulla oblongata, will exhibit their role and controls the respiration. So higher centers, I already told, there's a first centers and the major center. Quick response will be there. Rapidly, they can change the respiration, pattern of respiration, and will get back to the original or normal respiration from the higher or lower. Dorsal respiratory group will exhibit the normal respiratory manner, and it gives the signals to the ventral respiratory group, which will cause the expiration through the inspiratory muscle, which leads to the expiration. Neural mechanism. This is the neural mechanism. Okay, RCPG receive the input from PBC and abrupt increase in discharge. Dorsal respiratory group, which converts the RCPG into RAM signal. That RAM signal shut off by the phrenic control and 10th nerve from nerve. Paradoxical inspirations, signals coming from the FBC centers or from the VRG, paradoxical signals stops. Okay, so first two steps are inspiration and last three are expirations. So both inspiration and expirations controlled by the neural mechanism, which will be occurs in the brain. Paradoxical inspiratory signals, that is the signals for inspiration discharged during the end of the expiration so that breathing is smoother. Similar to a plane going noise up just before landing, so the landing is smoother. Okay, so the plane going noise up just before landing, so that the landing is smoother. Also, pertains to the second breath after delivery, that is 2 consecutive inspirations to inflate the lungs. Never again occurs during the life. So that is a paradoxical inspiratory signals. Higher centers, not essential for the normal respiratory activity. In case of the extreme low ventilation or extreme hyperventilation, it will place the important. Varied centers scatters through the nerves. Limited voluntary control. Upper motor neuron control to lower motor neuron of the respiratory muscle. Influences during the vascularization, deglutition, and playing wind instruments. Alterations during the emotional stages and peripheral chemoreceptor. The receptors respond to the change in the chemical composition in the blood or tissue fluid surrounding them. Basal firing rate depends on the normal mixture of the partial pressure of oxygen as well as the partial pressure of carbon dioxide. So, the respiratory center, peripheral chemoreceptor, triggered by increased partial pressure of carbon dioxide and increased hydrogen ion concentration. At the same time, decreased partial pressure of oxygen. And the receptor located to perform the chemoreceptors are carotid bodies and aortic bodies, which can detect the changes in the hydrogen ion concentration, oxygen, and carbon dioxide. These receptor gets stimulates. The receptor are known as carotid bodies and aortic bodies, which is located in the arch of aorta and carotid bifurcations. So whenever the changes in the blood carbon dioxide concentration or oxygen concentration or increased hydrogen ion concentration, these two receptors get stimulated and they send the signals to the hearing nerve, to the ninth nerve, and then into the respiratory centers. And aortic bodies, which is send the input to the 10th nerve. Finally, both the 9th and 10th nerve will convey to the respiratory centers. That respiratory center now modify or alter the respiration. Now, chemoreceptor cells are known as type 1 cells, known also called glomus cells, which contains dense organelles / vesicles and rich in catecholamines, responded by the dopamine release, cubed by the nerve endings. Type 2 cells, glial-like cells, supportive, nutritive to type 1 cells. So this will be a supporting cell to this type 1 as well as the type 2 cells. And blood supply to and metabolism will take place in this. 2000 ml of blood will flow per 100 gram tissues. Very high flow rate, extremely high metabolic rate, rapid build of anaerobic metabolites. Total tissue mass is 3 to 4 grams, and oxygen supply by the dissolved plasma oxygen. Stimulate to the peripheral chemoreceptor, increase partial pressure of carbon dioxide. When the blood carbon dioxide level increases, that is a highly sensitive to the chemoreceptor. It contributes to the 40% of the ventilatory responses. The response is de-innervated now. That is a decrease 30 percentage, and the increased hydrogen ion is parallels the increased partial pressure of carbon dioxide responses, and decrease oxygen pressure. See, previously the both increased CO2 and increased hydrogen ion is a potent stimulant to stimulate the respiratory center, and a decreased oxygen will be a less capacity to stimulate the respiratory center. The action exclusively on the peripheral chemoreceptors. Normal partial pressure of oxygen is 95 millimeter of mercury, and decreased partial pressure of oxygen to the 60 millimeter mercury, gradual and proportional increase in the receptor activity, but no ventilatory changes. The following three factors or following three changes will happen during this mechanism. Number one is the upper plateau of the hemoglobin oxygen curve, and deoxyhemoglobin weaker acid than the oxyhemoglobin. Therefore, the hydrogen ion in the plasma, and if increased ventilation decreases the partial pressure of carbon dioxide, leads to the decreased respiratory drive. Then 60 to 30 millimeter of mercury hyperventilation, which leads to increased rate and increased depth of the respiration, both, and steep portion of the hemoglobin oxygen curve. De-innervated chemoreceptor, which leads to the paradoxical decreased ventilation in the hypoxia and minimal pH variability responses, which shows that the primary stimulus to the peripheral chemoreceptor is to increase the partial pressure of carbon dioxide and not decrease the partial pressure of oxygen. Increased potassium, that is an example for excess hyperventilation and venous stasis of anything which will cause the oxygen delivery is reduced. Cytotoxic agents, cyanide, which leads to inability to utilize the oxygen. and nicotine by raising the carbon monoxide and carbon dioxide level. Next, central chemoreceptor. The chemoreceptor is located in the central nervous system as follows: ventral lateral surface of the medulla, hypothalamus, nucleus of tractus solitarius, locus ceruleus, etc. More dominant and powerful than the peripheral chemoreceptor. So the central chemoreceptor more dominant and powerful than the chemoreceptor. That is, in percentage wise, it will be around 75 to 80% of the total responses. Stimuli to the central chemoreceptors increase hydrogen ion in the cerebrospinal fluid. Number one, CO2 easily crosses the blood-brain barrier into the cerebrospinal fluid, and rapid hydration and dissociation leads to the hydrogen, and hydrogen does not cross the blood-brain barrier. CSF protein negligible and minimal buffering. Thus, small change in the PCO2, large changes in the CSF pH. No carbonic anhydrase in the CSF, therefore reaction will be slower. Also acts directly on the respiratory centers and increase partial pressure of carbon dioxide as follows: blood hydrogen normally buffered by the sodium bicarbonate, thus acidemia has little effect. In chronic states, kidney excretes the sodium bicarbonate. Deep and rapid breathing, Kussmaul's breathing seen in the diabetic ketoacidosis. This is a flow chart which shows the regulation of all the pattern in our body happens, which will be totally controlled by the brain respiratory center. The chemosensitive area is a specialized area present in the brain, which will be only the area will respond to the changes in the hydrogen concentration. The other respiratory centers like DRG, VRG, Abnist center, pneumotaxis center will not respond to the changes in the blood hydrogen ion concentration. Okay, the hydrogen ion highly sensitive to the chemosensitive area. Whenever the changes in the hydrogen concentration, it will get sensitized and give the signals to the respiratory center, and accordingly the respiratory center will modify or alter the respiration. Lung reflexes. The location of the lung reflexes, the present in the lung parenchyma and air passages as well as in the blood vessels. So these lung receptors are slowly adapting and rapidly adapting. Now supply vagus nerve, that is the 10th nerve, small diameter myelinated fibers, A delta fiber, and unmyelinated nerve fiber, type C fiber. Slow adapting, receptor activity persists as long as the stimulus. The receptor airway smooth muscles, A delta, which will monitor the extent of the inflammation, extent of the inflation and deflation of the lungs. Response modify the expiratory pattern. Associated responses are bronchodilation and tachycardia. Hering-Brewer reflex, that is a two main reflexes: Hering-Brewer reflex, Hering-Brewer inflation reflex, as well as the Hering-Brewer deflation reflex. Increases the duration of the expiration when the inspiration is excessive. Deflation reflex increases duration of the inspirations when the expiration is excessive. Neural centers are pneumotaxic, amnestic, dorsal respiratory. Rapidly adapting, lung irritant receptor, location airway epithelium. These receptors are very fast adapting, and stimuli for these receptors are chemically dust, gases, and smoke, and marked inflammation and deflation of the lungs, pneumothorax, as well as the pulmonary congestion. Afferences for these rapid adapting receptors: small myelinated vagal fibers, and responses are hypernia and bradycardia. Clinically, might be responsible, example, asthma. Lung J receptor which is discovered by the scientists Painton, location alveolar walls close to the capillaries. Stimuli for these receptors are increased interstitial fluid volume in alveoli and pulmonary congestion, edema, and microemboli. Afferents are for these juxtamedullary receptors and myelinated vagal fibers, and the following responses will occur as bronchoconstriction, shallow and rapid breathing, and decreased heart rate and decreased blood pressure. Clinically, dyspnea of the heart failure. And other reflexes are carotid sinuses and body baroreceptor, which will be controlled from the cardiovascular system, and skin sensations, proprioceptors, and higher centers, that is a central nervous system. Thank you.